Hello, this is now starting of the session number 3 of the sector molecular basis of inheritance for the second year students. Already I have described about the chemical contrast between DNA and RNA. RNA or DNA chemical structure. Schooling or posting of our GN hot like a difference to follow. Difference between RNA and DNA I have described in my last session, session number 2. Now that will deal with the formation of DNA and RNA. How this nucleotides or the concept of nucleotides already you have learned. Nucleotide chemical form I can call but repeating again the same concept. My repeat also repeating about the same concept how a nucleotide is formed. As I have described, a nucleotide is formed by three kinds of molecules. Therefore, a nucleotide is called a trimer. You can write trimer. Three type of molecule. There is a nitrogenous base, there is a pentose sugar, and there is a phosphate group. This is true for both DNA and RNA. RNA, DNA, duita karne a concept to applicable hai. But again, DNA thoka nucleotide bami how ki boli kaun deoxyribonucleotides boli kaun. You note that you reminded the nucleotides of DNA are characterized by the presence of the pentose sugar deoxyribose. But again, this is the sole feature on which the deoxyribonucleotides are distinguished from the RNA nucleotides or ribonucleotides. RNA nucleotides contain ribose sugar, DNA nucleotides contain deoxyribose sugar. It was first difference, chemical difference of the second difference of in DNA, the purine bases are adenine guanine present, and this is true for RNA as well. RNA to hit a purine base adenine guanine. Or do that purine base. Pyrimidine base happy difference of the purine base. There is a difference we find between RNA and DNA. The difference is overall in nucleic acids means RNA and DNA combined together. 3 types of pyrimidine bases are present. These pyrimidine bases are cytosine, thymine, and uracil. Out of these 3, only 2 present in DNA and only 2 types present in RNA. DNA থকা pyrimidine base kita hol thymine aru cytosine. RNA থকা pyrimidine base kita hol uracil aru cytosine. Gotike samna ami ki paluge base apekhe reference to bases DNA and RNA differs by pyrimidine bases only, not in the pyrimidine. Mane difference in base composition in DNA and RNA occur with reference to the pyrimidine bases only. Pyrimidine base low hai ami RNA or DNA matter difference pa. Accept hoise I think. No, you have accepted about this concept I think. So accept hoise etu. Tia ki paluga ami a nucleotide is a trimer, whether it is a RNA nucleotide, ribonucleotide or deoxyribonucleotides. A nucleotide is always a trimeric molecule. Hoday a trimer molecule. Gotike structurally, how a nucleotide is created. Nucleotide can form. A nucleotide is created by a formation of by 3 kind of molecules. Here sugar to a central molecule. Nucleotide in a nucleotide, the central molecule is a sugar. The nucleotide to DNA RNA doesn't matter. Central molecule to sugar to right hand dot. There is a nitrogenous base, and on the left side there is a phosphate group. This is a diagram presentation for a nucleotide. What I can do? Because DNA side on nucleotide pound, RNA to side on nucleotide pound. Deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate are the nucleotides of DNA. And similarly, adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and uridine monophosphate are the nucleotides of RNA. You see, DAMP, DZMP, DCMP, DTMP are the nucleotides of DNA. And likewise, AMP, ZMP, CMP, and UMP are the nucleotides of RNA. RNA nucleotide, DNA nucleotide. What I would tell you? এই নিউক্লিয়ৰ কিটা প্ৰতিটো নিউক্লিয়ৰ মানে চাৰিটা মলিকুল থাকে প্ৰতি নিউক্লিয়ৰ কিন্তু মাজৰ সেন্ট্ৰেল মলিকুলটো হ'ল সুগাৰ the central molecule in a nucleotide is always a sugar central molecule is always a sugar the sugar if I draw a sugar chemical structure of a sugar suppose it is a deoxyribonucleotide or drawing deoxyribonucleotide suppose it is if I draw the structure of a deoxyribonucleotide এটা structure যদি মই draw কৰো deoxyribonucleotide structure যদি draw কৰো সেটো deoxyribonucleotide central molecule to sugar right handed at a base or left handed at a phosphate this is a diacritic sketch for a nucleotide structure number sugar to যদি nucleotide to deoxyribose মানে DNA nucleotide যদি হয় if the nucleotide belongs to DNA then sugar will be deoxyribose sugar so we have drawn the structure of the deoxyribose sugar this way it whole deoxyribose sugar structure this is the skeleton of the deoxyribose sugar okay this is the position number 1 occupied by 1 carbon position number 2 occupied by second carbon 3 carbon 4th carbon and 5th carbon 5th carbon contain a primary alcohol group CH2OH and there is also a hydrogen atom there is an H at the position number 3 also H OH here but A's here OH okay it actually is the structure of beta deoxyribose sugar beta deoxyribose or simply DR deoxyribose it was central molecule so position number 1 carbon 1 carbon 2 carbon 3 carbon 4 and this is carbon number 5 this is carbon 5 okay the carbon 1 2 3 4 5 it has cytocarbon carbon deoxyribose sugar of structure it is the first carbon containing the hydroxyl group above is actually beta form tamane it whole beta deoxyribose sugar but it is a structure of beta deoxyribose mane nucleic acid at thoka sugar like beta form again not here the sugars pentose sugars present in nucleic acids are in beta form kile thake on hydroxyl group carbon what this is the sugar at top this base to suppose my draw here this is the base suppose adenine base to sugar adenine now I am drawing upside down tamane pentose moiety to my equation pentose moiety to my equation okay now it will nitrogen this is nitrogen this is carbon here this is carbon this is carbon this is carbon this position is nitrogen this position carbon this position nitrogen there is an H here there is a double bond adenine jodi hoy etu thikibo NH2 another econo thake C at a H double bond okay etu hol adenine base etu adenine okay yet eta thake H nitrogen 9th nitrogen 1 2 3 4 5 6 7 8 9 nitrogen because this OH and this H will join together and eliminate a molecule of water okay water molecule eliminated in this process of elimination of water is called dehydration takes place between carbon number 1 of deoxyribose sugar and the hydrogen attached to the N9 of the purine base adenine okay dehydration lead to condensation dehydration lead into condensation dehydration lead into condensation that means water molecule eliminate kori OH to গুচি গ'ল ইয়াৰ পৰা আৰু ইয়াৰ H to গুচি গ'ল H and OH are standoff and so that the N bond will continue directly with the C1 of the sugar forming total গুচি গ'ল to dehydration okay forming means the two as a result combines deoxyribose sugar bounded to the N9 of the purine base adenine by one bond this CN bond we want to hold C1N9 bond this C1N9 is going to be called glycoside bond C1N9 it hold glycosidic bond by forming a glycosidic bond between N9 of the purine base and C1 of deoxyribose sugar, a dimer is created prior to formation of a trimer. Means nuclear form hoga nai. Nuclear form hola sugar to phosphate ko logai hoye. Prothomte base to sugar logai jano then sugar to base to eta phosphate logai. Is the dimer component palo sugar plus base. Sugar plus base this component dimer is called a nucleoside. Nucleoside will be called. Okay. The dimeric molecule that is formed prior to formation of a trimer nucleotide is called a nucleoside. So definitely that there will be 4 kinds of nucleosides. The nucleoside formed between adenine and the deoxyribose sugar in DNA is called deoxyadenosine. Kidney monophosphate monophosphate since phosphate group is not added till now, it is a nucleoside. But deoxyadenosine is the example of nucleoside. The nucleoside or example. Deoxyadenosine is an example. Here look at the phosphate group of a tide. So this is base plus sugar plus phosphate group. The deoxyadenosine monophosphate is the phosphate group. But it's not a nucleoside at a person. This is number 1 information you are getting. Then next, with the sugar or a 5 carbon to look at hydroxyl group. So the hydroxyl group attached to the 5th carbon of the deoxyribose sugar in DNA will join with phosphate group. Phosphate group structure of phosphate group can be represented like this way. It was phosphate group. Okay. Phosphate group or a hydroxyl group are 5th carbon of sugar to it hydroxyl. The two will join together and eliminate again a molecule of water. This way. Here water molecule again 1 molecule of water will be eliminated. And this is again a dehydration process. This is again dehydration. Water molecule possible. Dehydration lead into condensation again. Condensation. But again, this water molecule is eliminated by joining this to a O to this O will be left. Sugar to base O to this O is left of the sugar and start to join the phosphate group. It is still a phosphoric acid. It was phosphoric acid or phosphate group is originally phosphoric acid molecule. Originally this is phosphoric acid. Whatever we call it, phosphate group phosphoric acid phosphate group hisabe thake living system phosphoric acid or phosphate group bounded to the 5th carbon of the sugar again by acid and into alcohol acid plus acid alcohol and a bridge create. The bridge to become ester bridge, ester bond. Ester bond. Kintu jehetu acid to phosphoric acid hoy gotike ইয়াক আমি কওঁ phosphoester bond. Phosphoester bond. The bond created so is called phosphoester bond. Phosphoester. This is phosphoester bond. Phosphoester bond. Phosphoester bond is created. Phosphoester bond. There will be double condensation in order to form a nucleotide in a nucleic acid. a nucleotide is a double condensation product among sugar, nitrogenous base and a phosphate group. A nucleotide is a double condensation of how ultimate conclusion can be made this way. Ultimate conclusion will be like this. Ultimate conclusion that a nucleotide is a double condensation product among sugar, base and phosphate group. Okay? Double condensation or double dehydration. Double condensation okay? So these are nucleotides are created. Keep on getting it. It is whole nucleotide structure. You go by this one, emulsification. To one the first question in a nucleotide note is into molecule. Note in a nucleotide, irrespective of the type of the nucleotide type 0 in a nucleotide, a central pentose sugar remain linked with essential sugar remain linked with a base by a glycosidic bond and also the same sugar remain joined with same sugar remain joined with a phosphate group by a phosphoester bond. These information are very important from competitive exams point of view. Competitive exam important. But definition nucleotide is a double condensation product among sugar, base and phosphate group in which the sugar remain linked with the base by means of a glycosidic bond and that of the phosphate group by means of a phosphoester bond. Okay? যদি বেসটো পিউরিন হয় তেতিয়াহ'লে গ্লাইকোসাইডিক বন্ড হ'ব C1N9 বন্ড। If the base is purine, যদি বেসটো পিউরিন যদি বেস পিউরিন হয় তেতিয়াহ'লে সুগাৰৰ ফাৰ্ষ্ট কাৰ্বনটোৰ লগত বেসটো 9 নাইট্ৰজেনৰ লগত বান্ধনি থাকিব মানে গ্লাইকোসাইডিক বন্ড থাকিব C1N9 bond. Glycosidic bond will be C1N9 if the base is purine. যদি base to purine হয় তেতিয়াহ'লে glycosidic bond between sugar and the base will be C1N9. Next molecule, if the base is pyrimidine, যদি base to pyrimidine, if base is pyrimidine type, then the bond will be C1N3 bond. Or C1N1 bond. C1N1, obviously there is a controversy present. Yet at a controversy C1N1. There are two types of glycosidic bonds present in the nucleotides of DNA, RNA combined liver and nucleic acids. C1N9 if the base is purine, or C1N1 or N3 if the base is pyridine. But this information is doubtful. Some describes this is C1N3 and some describes this C1N1. So that will be your choice. C1N1 likha, C1N3 likha, কিন্তু C1N9 at least লিখিব নোৱাৰা। Pyrimidinous base to what like glycosidic bond over C1 and 1 by N3. Pyridinol C1 and 9. এইটো হ'ল নিউক্লিওটাইডৰ ডেফিনিশ্যন আৰু এই ইনফৰমেশ্যনটো তোমালোকে মই ৰাখিবা লাগিব গ্লাইকোছিডিক বন্ড এণ্ড ফসফোএষ্টাৰ বন্ড হয় কি গ্লাইকোছিডিক বন্ডৰ দ্বাৰা চেন্ট্ৰেল সুগাৰটো বেছৰ লগত লাগি থাকে আৰু ফসফোএষ্টাৰ বন্ডৰ দ্বাৰা চেন্ট্ৰেল সুগাৰটো ফসফেট গ্ৰুপৰ লগত লাগি থাকে ইন কেছ অফ নিউক্লিওটাইডৰ ক্ষেত্ৰত ওকে so this is information therefore এইটো হ'ল নিউক্লিওটাইড নাম্বাৰ 1 গতিকে একে ধৰণে মই বাকী ষ্ট্ৰাকচাৰ ধৰণৰ কৰোঁ তোমালোকক by same mechanism nucleotides are created. This is the nucleotide so created. Yet to have base to adenine deoxyribose sugar phosphate group adenine DRP ducation deoxyribose monophosphate structure deoxyribose monophosphate DAMP. This is the structure of DAMP. Structure of DAMP molecule it is deoxyribose monophosphate. Okay. What is the structure drawn up over? Nucleotide can have formally follow. Look, let's look here. Nucleotide structure. This is a compare. This is internet download product. Deoxyribose sugar is this, phosphate group is this, adenine base is this. The adenine base are sugar, AGD bond, roll glycosidic bond. Deoxyribose sugar, phosphate group, AGD bond, E bond to her, phosphoester bond. Okay. This is adenosine monophosphate structure of adenosine monophosphate. Okay, now this is adenosine monophosphate structure of a typical nucleotide. Next part, these nucleotides are 4 types. Okay, diagrammatically the 4 types of nucleotides present in DNA can be represented as under. Diagrammatically there. Okay, now students, I am continuing about the structure of nucleotides. I have found that in DNA there are 4 different types of nucleotides present. These 4 different nucleotides can diagrammatically be structure. I have given one. Remember, the structure of the nucleotides individually I have given, but only one structure I have given. That structure was AMP, DAMP structure I have given, but nucleotides can be represented diagrammatically as well. Nucleotide diagonally can be represented. Always you remind, deoxyribose sugar is present as the central molecule. On the right hand side there is an adenosine base. Suppose the base is adenine, and on the left side there is a phosphate group present. There is present a phosphate group. Okay, this is nucleotide number 1. Now this nucleotide containing adenine base is called adenosine monophosphate because the sugar is deoxyribose sugar. It is deoxyadenosine monophosphate. But this is actually diagrammatic presentation of deoxyadenosine monophosphate. It is called deoxyadenosine monophosphate or diagrammatic sketch. In DNA, as we have already described, there are 4 different types of nucleotides present. These 4 types of nucleotides are diagrammatically therefore presented. Again, deoxyribose sugar. Suppose next one is cytosine base present. The next base is suppose cytosine, and phosphate group is suppose this is phosphate group P I am writing. Next nucleotide, this is deoxycytidine monophosphate. Next nucleotide is deoxyribose sugar, guanine base phosphate group. Next nucleotide phosphate group, deoxyribose sugar, and say it is thymine. Since in DNA the thymine nucleotide is present, means thymine base is present. So this is deoxyguanosine monophosphate, and this is deoxythymidine monophosphate, DMP. Deoxyadenosine monophosphate, deoxyguanosine cytidine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate. These are 4 nucleotides, and these 4 nucleotides are alternately arranged. Maybe adenine followed by cytosine, then guanine or thymidine, maybe, or adenine followed by guanine, cytosine, thymine, or adenine, thymine, guanine, cytosine, maybe any series. But all these 4 nucleotides must be present in the DNA molecule. So a DNA is characterized by therefore 4 nucleotides. 4 types of nucleotides are present. But you remind, in a DNA molecule, these numbers of nucleotides present vary according to complexity of the organism. The simpler the organism, the lesser will be complexity of the or length of the DNA nucleotide chain, polymerase chain. The complexity of the organism if the complexity of the organism is more, the chain length will be more. So variability of the chain, complexity of the chain depends on the length of the DNA chains. You remember. Okay, but again note, in a nucleotide chain, actually nucleotides serially arranged one by one, they form long polymerase chains. Okay, but in a DNA or RNA molecule, the nucleotides never occurs independently. They must be united with the successive nucleotide. So their joining occur between the 2 nucleotides, adjacent nucleotides. The junction between 2 adjacent nucleotides takes place via the deoxyribose sugar of the previous nucleotide with the successive phosphate group of the next nucleotide. Means deoxyribose sugar of the first nucleotide, the phosphate group of the second. The deoxyribose sugar of the second nucleotide with the third phosphate group of the nucleotide. Means second deoxyribose sugar nucleotide, second deoxyribose sugar of the nucleotide with the third phosphate group, third deoxyribose sugar with the fourth phosphate group, and so on and so forth. So this way, in a DNA chain, the phosphate groups, one phosphate group of a nucleotide have to join 2 nucleotides to each other. The junction between the 2, actually, as I have described, in deoxyribose sugar, there is a 3' hydroxyl group remain free. 3' hydroxyl group always remain free. 3' hydroxyl group always remain free. 3' hydroxyl group always remain free. And in a nucleotide, ordinary nucleotide, the phosphate group remain attached to the 5th position of the sugar. 5th position of the sugar, 5th position of the sugar, 5th position. So the phosphate group of the first nucleotide from the 5' end of the DNA, and the last deoxyribose sugar where there is no further attachment of the phosphate group will form the 3' hydroxyl group. So this end, starting end of a nucleotide in the chain can be designated as 5, and the ending end, last termine is designated as 3' end. So there is a direction found in the DNA molecule running from 5' to 3' end. This is so-called the polarity of the DNA molecule. This is called polarity of the DNA chain. Polarity of the DNA chain. But you remind one thing, DNA in most cases is double stranded. This is only one strand. The other strand run in antiparallel direction, that's what I will do later on. So there is a polarity. Every strand of the DNA, every polymerase chain of a DNA molecule is, it bears polarity from 5' to 3' and we discussed this way. This strand can be 5' to 3' like this way. Okay, this polarity always exhibited by DNA molecule. So, but independently, nucleotides never occur independently, as they must unite. The union occurs between the sugar of the previous nucleotide with the subsequent phosphate group always. The phosphate group of the subsequent nucleotide contain always an H group pointing towards the hydroxyl of the previous nucleotide like this way. Okay, so 3' hydroxyl of the previous sugar's nucleotide of sugar reacts with the hydroxyl group of subsequent phosphate group and eliminates a molecule of water through dehydration or by condensation. The actual dehydration of elimination of water is called dehydration. Okay. As a result, condensation takes place. That means one nucleotide joins with nearby nucleotide in a chain by means of the sugar phosphate bond. This sugar phosphate bond again called phosphoester bond. এই বন্ধটো আমি আকৌ কি বুলি কম phosphoester bond বুলি কম phosphoester bond আৰু সেই ফসফেট গ্ৰুপ যাৰ লগত প্ৰিভিয়াছ সুগাৰ জড়িত তাৰ লগত নিজৰ phosphoester bond থাকে again there is a phosphoester bond present also phosphoester there will be therefore two phosphoester bonds by which the phosphate group serve to link adjacent nucleotides উচ্চাৰণকৈ থকা দুটা নিউক্লিওটাইড সদায় একেটা ফসফেট গ্ৰুপৰ লগত লগ লাগি থাকে আৰু এই বন্ধটো হ'ব ফসফোডাইএষ্টাৰ বন্ধ এই বন্ধটো this bond we can call as phosphodiester bond. This is so-called phosphodiester bond. Phosphodiester bond. Phosphodiester. So again, this is phosphodiester bond. This is phosphodiester bond. So what is ultimately we are obtaining that phosphodiester bonds serve to join adjacent nucleotides together and forms the nucleotides, the DNA nucleotides this way. So one nucleotide formation is shown here. The next nucleotide also formed, but it runs opposite direction. So next nucleotide will run in opposite direction, forming two antiparallel chains. This overall, my objective up to this in session 3 is to describe about how a polynucleotide chain is created, whether it is DNA or RNA. So further description of the next subsequent parts of how DNA molecule is formed, the double helical model of the DNA molecule, and many more information that I have to give, I will give in the next class. Okay, thank you.